Good morning, good afternoon, good evening, wherever you are. Thank you for joining the Clarity webinar. Today is the first of a series. My name is Lewis Bond. I am the chairman of the ReSVINET Foundation and part of the Clarity Consortium. It is a great privilege today to chair this webinar. Clarity has the mission to generate preventive and therapeutic approaches for chronic respiratory diseases with the vision that knowing the molecular mechanisms how infections change the airway will enable us to develop preventive or therapeutic interventions, with the final goal to impact the understanding, prevention, and possible treatment of virus-triggered asthma pathogenesis. Today, we talk about from RSV to asthma, unraveling the link. Again, it's the first of a series of webinars that you can expect from Clarity. In the course of the webinar, we invite you to ask your questions in the Q&A chat. It's very helpful for me, understanding your questions beforehand, so then I can accommodate your questions during the Q&A session, for which we will have plenty of time. So who are these speakers? Professor Dr. Marianne Boes is an immunologist who is the lead of the Pediatric Immunology Department at the Center for Translational Immunology of the University Medical Center, Utrecht. She is the coordinator of the Clarity project. After many years working at Harvard Medical School, she came back to Utrecht here in my department, where I also work, to help understand various diseases from a mechanistic point of view, which has led to tremendous amount of insight in various diseases. The second speaker is Professor Dr. Pascal Walter-Braun, who is a trained biochemist and director of the Institute for Network Biology at the Helmholtz Institute in Munich, Germany. And he basically calls himself now a systems biology expert, trying to use statistical approaches to understand diseases. Without further delay, I would like to wish you a great webinar and remind you again that you should use the Q&A chat button for your questions in the course of the meeting. With that, I give the floor to Marianne. Marianne, take it away.
Microphone.
Thank you so much, Louis, and thank you, Pascal, for reminding me to use the microphone. Thank you for this really kind introduction. So I'm talking from University Medical Center in Utrecht, and I'll start this webinar, at least this part of the webinar, by explaining a little bit how we came to study the role of RSV, respiratory syncytial virus infection, in later development of asthma or other chronic lung diseases. Now, the connection between RSV and asthma is quite well known. If we look on the internet at the normal websites, not peer-reviewed journals, we already find pages like this exploring the connections between RSV and asthma. Now, in peer-reviewed journals, we look at PubMed. I think many of the people listening in today use PubMed to look at peer-reviewed journals. We see since the 60s or 70s already, people are investigating the connection between RSV and asthma exacerbation or asthma development. There's a ton of papers out there. And I will explain a few of these examples. Already in the Lancet in 1999, there's a commentary on RSV in early life before the age of 1, how in some children This increased the risk of wheeze and allergy or asthma by the age of 13. There's a paper in Thorax in 2010, 10 years later, 12 years later. When you look at the bottom, when you have a parent with asthma, you have a slightly increased chance of developing asthma later in life. And this is after 18 years. But when you now also have had RSV bronchiolitis, so severe infection with RSV virus early in life as an infant, then you really increase the risk of developing asthma as an adult. The same is true for allergic rhinoconjunctivitis, so another allergic disease. So somehow this early event, RSV infection in the lungs, increased this risk in certain predisposed individuals. Lancet again, 2023, really reputed journal, again emphasizing that there is an increased risk of developing asthma when you've early on in life, as an infant, contracted severe RSV infection in your lungs. Now in Utrecht, we've been investigating this for a long time. Louis, actually, who's now a dean in our hospital, he actually studied this as a PhD student, and also his supervisor in 1992. This is almost, well, is it 35 years ago?
1998.
1998? No, but Jan Kimpert from 92. His first paper I noticed. No? I think Jan already worked on this.
Jan was way before me. Go on.
There you go. So a severe RSV infection really causes this inflammation in the lungs and somehow triggers later on development of asthma. But early on, we only investigated these early events, the severe acute infection. And with Louis, this is a bit later, 2014, 2013, the two of us teamed up and we investigated the role of IL-17 cytokine together with IL-8 induces the influx of neutrophils in the lungs of these children who are intubated and in the neonatal intensive care. We found that in these children, 5 hours or 48 hours after they are admitted in intensive care, they already have increased levels of IL-17, also IL-8. But really there's this connection between IL-17 and neutrophil presence in the tracheal aspirates, so these airway fluids. What we next did, we looked in mice that we infected through the nose with RSV. And we did that in the presence of antibody that blocks IL-17 or not. We did it with a low plaque-forming unit, low dose of virus or high dose of virus. You see fewer cells when you block the IL-17 from working. So fewer influx of cells. And these cells are neutrophils. Really, this is blocked by presence of IL-17A. We followed up. And we really looked at the presence of IL-17 again in these lungs. And we looked at what induces the polarization of CD4 T cell responses towards Th17, that they start to produce IL-17. And that's really the production of cytokines IL-6 and IL-23 by the professional antigen-presenting cells in the lungs, local. And these are the macrophages and the dendritic cells. They really educate the CD4 T cells to become Th17 cells. And there's a connection between IL-17 producing cells and Th2 responses in development of later asthma. And this is really what we wanted to do, but it took again another 10 years before we got to do this. Now, we're not the only ones studying severe RSV infection in how it relates to later asthma development. There's a lot of people studying this. Maybe some of them are in the audience. And we see that there's a positive connection between severe RSV infection and when you're born at preterm age, or there's a viral co-infection, low birth weight, being male. So if it's a co-infection, or a parent having asthma already. And there's things that protect. For example, a passive or an active vaccination against RSV, other vaccinations. We thought that's a lot of information to process. We take the genetic approach. And we tried to come together. We found this team. Pascal Valter-Brown will be speaking in a bit. We found other team members to together make the project Clarity we're discussing today. We look at the genetic elements in RSV viruses and the genetic elements in the host that later combine to develop, to cause the development of asthma. I would like to give the floor now to Pascal, who will explain the details, the nitty-gritty of this project.
Thank you so much, Marianne. Also from me, welcome, and thank you all for being here today. And I will just pick up where Marianne left and briefly explain what we try to do in the Clarity Consortium, and then also how we want to bring together the viral and the genetic component in this fairly unbiased approach that we start from. So Marianne already explained that both RSV and human genetics impact the development of asthma and allergies. And so they are coming in an infection, they are coming together in that specific RSV strain influences or infects an individual that has a very specific individual predisposition. And now there's a decision point, and some kids get only a moderate infection, whereas others get a very severe infection. And then for these ones, there's a much increased risk of developing allergies and asthma. And so we want to understand the determinants of both what determines if an RSV infection results in such a severe course of the disease, and then also what determines what is happening later in life. And we've structured our consortium in which we are working and addressing these questions around these three questions. One is, what are the factors that influence infection severity and asthma development? And this is not only, it's somewhere genetics, but it's a more precise question. In a second question, and this is what I want to spend the bulk of this presentation on, is how can we learn how these factors synergize on a molecular level to influence infection severity and also the asthma and allergy development later on. And then the last part that Marianne will then take the floor back is how can we use this understanding to identify the molecular mechanisms and use these mechanisms for therapy and for preventive approaches. So let's first talk about the genetic side. Many of you may know genome-wide association studies. In this particular approach, we look at a large population, large numbers of individuals who had, for example, in this case, a severe course of an RSV infection or a mild course of RSV infection. And these are then genotyped. So we're determining the specific genetic variants that they have at a variety of loci. And from this, you can then do statistics and you identify individual positions in the genome that have a variant that increases the risk. And this is a work that is being done by colleagues at the University of Tartu, at the Estonian Biobank, which have genetic and health records for about 15% of the Estonian population. And from this part, we expect to get information on which genetic risk factors exist and influence the outcome of an RSV infection and the later development of asthma and allergy. The second aspect that we look at or that we want to find out in terms of what are the factors are this particular RSV strains that may lead to a more severe course of an infection or not. And we all know from the pandemic that we've gladfully enjoy and fortunately passed by now to at least the most severe part, that viruses are constantly developing. There's new variants that are emerging. They have slightly different both immunologic phenotypes, but also what they're doing inside the cells. And our colleagues at the National Center for Microbiology in Madrid, they have a very long, they are also the reference lab for respiratory viruses. and they are analyzing viral populations and doing the full genome sequencing, relate this back to the clinical data in order to identify and build again correlations between what viral subtypes tend to lead to more severe infections. And so we get information from both sides. On the one hand, the host genetics, and from this part, which viral strains are potential risk factors for the phenotypes that we care about. So these parts that may be covered in future webinars in this series give us information on human genetics and particular strains. And what I want to talk about now, how these factors, how we want to study and try to find out how these factors come together on a molecular level. When I mean molecular level, I want to briefly start from the bird's eye view and what we see in our daily lives. We have an individual, and this may or may not get infected, hopefully not, but if it does, it's usually the lung that is being infected. And so in the lung, it's not the lung as a tissue, but in the lung, of course, consists of individual cells that make up, in this case, the epithelial layer. And this is a histological image we can also have and represent this as this cartoon where we see different cell types. In each of these cell types, we do have very complex molecular networks. And it's these molecular networks that are both the subject of genetic variation. So here we see small changes in between us. All of us have individual variants that lead to small changes in how these proteins that we're looking at, what they look like and how they're behaving. And at the same time, the viruses enter these cells and replicate inside these cells. So they're perturbing and subverting these molecular networks and synergize and act together here with the genetic variation. And this is what we want to understand. So in brief, we have the virus, we have human genetics. We want to understand how it influences the lung and disease outcome. But in order to do so, we need to look at these. We have to look at these molecular networks. And because this can be sometimes very abstract, I want to take a little path to the side and explain how we study molecular networks or networks, complex networks, in different contexts. So in our case, we want to look at molecular protein networks, protein signaling, protein-protein interactions, and we can represent them as these very complex networks that basically mediate cellular behavior. But complex networks also exist in other contexts, and they have a very striking similarity to these protein networks. And so one example that I want to comment on are these social networks. We all interact in these social networks, and by now we can also visualize and formalize them on these diverse platforms. And people have been starting to look at these networks already for 20 years and exemplified and tried to use these to learn something about this. And we all know these platforms, as well as national intelligence agencies and spy agencies, they spend a lot of money investing in these networks to learn something. And so I want to give you a little glimpse of what we can learn from these networks and how we relate this back to biology. This is one example from a high school social network that was studied more than 20 years ago. And in this network was constructed by asking the students if he or she is a friend, if they consider someone else a friend. You can see there are some loners and some that are highly popular in the center. But what we can also see that there are these 4 clusters, 4 groups. And the question is, what's going on there? What can we learn? What are these groups? And when we look at some of the underlying data, we realize one of those is a perceived ethnicity divide, right? So we have kids with a different appearance, self-identified ethnicity, and this is a major division here, and that is very apparent in the data. And the second divide, maybe also not so surprising, is a gender divide, that guys and girls tend to be more friends with each other. An important consequence out of this, however, is if one of the students agrees to say, yes, I'm telling you who my friends are, but I don't want you to know how I identify as a gender, and I don't also want to identify as my ethnicity. Just from the position in the network, we can have a very, very strong hypothesis about what this person looks like. And this is not only about outward appearance, it goes even deeper. And this is 20 years ago, but everybody knows this co-bias network, these recommendations. Other people who bought this item also bought the following thing. And these data are being analyzed. And when you analyze this about political books like 20 years ago, you realize that there are major divisions. Again, these clusters, these communities that are emerging. We saw this in the first example. We see this again here. They have a certain color here. But if we now look behind the scenes, what are these kind of books? We realize that some of them are much more conservative or much more liberal. So from the data and from the traces that we leave online, there are very deep and profound insights that the institutions and the companies can draw and learn about us individually on a very personal level. And so I want to do, this is from 20 years ago. The possibilities today and nowadays go much, much deeper. But our topic here is I wanted to use this to illustrate. Our topic is how we can use this to study human disease. And in human disease, we have a very similar phenomenon. We can identify through genetics and network and genome-wide association studies, we can identify loci in the genome, and they tend to be very dispersed. But when we now look at them on a molecular network and look at their connectivity, and we can see, as in this example on ataxias, which is unrelated to our topic, that they're often very closely together. And again, they form these modules and these communities that act together and do something that we can identify. And what we want to do in our project is to not only identify those, but we want to identify how the asthma modules are targeted by viral proteins and learn about the relationship between viral proteins, genetic predisposition, and how they influence the cellular behavior and features. The way we aim to do this, I want to illustrate with a study briefly with some results that we have collected in the context of the SARS-CoV infection. Because here you may remember that we also had a lot of different diseases here circled and features and phenotypes that were implicated and linked to a severe course of an infection. We have thyroid disease, body mass index, and so on. And so the question is, how does this relate to the actual infection? How does it relate to what the virus is doing? And there are different models. So we knew already that there are some loci involved and there is certain genetic variants that increase the risk for a severe course of disease. So we have these genes and encoded proteins, and we know what these, and we know that these can increase the risk of an infection. What we didn't know is if these ones, and these are two hypotheses, if these ones are directly targeted by the viral proteins, or if they may be indirectly targeted, and these are some sort of network effects. And this was what we wanted to address in this particular study. So we started with the host network, and so all the proteins and protein interactions that we carry inside of our cells, that are going on inside our cells all the time. And we first asked, what are the what proteins of the human host are binding to these viral proteins? What are the targets of these viral proteins? And so we do have an experimental platform to address this question. And in the second step, we then asked, okay, now that we know the viral targets and we know the genetic variation, how did these two relate to each other? And we saw very clearly that there was never a viral target, one of these severe COVID genes that was targeted directly, but they were always indirect. So they were indirect, one hop away of the viral protein. This is targeting one protein, and then one of the interaction partners is involved in severe COVID in this case. Moreover, what we found is that when we look at the genetics for other important comorbidities, they're also found in these communities. And so when we did this in a statistical and unbiased way, we find also susceptibility genes for an increased body mass index, or for thyroiditis, hypothyroidism down here, and schizophrenia, other traits that had been linked to severe COVID. And so what I want to say here, what I want you to understand is that we can use these networks to link genetic risk factors to biochemical mechanisms and to what the virus is doing. And we could then actually go and follow up on this. We demonstrated that these viral targets in mechanistic assays, that these viral proteins are actually activating the signaling, that when we knock out the human targets, they can sometimes severely impact viral replication. And we can also use this to find small molecules that interfere with some of these things that the viruses are doing. And this is the general approach that we now want to use also in the Clarity project to bring together RSV infection, or what the RSV viruses are doing, and what the genetic variation is doing. So in our project now, we start with viral proteins, a little bit more than 200 proteins of 15 different RSV strains. And we do the network mapping just as as I've just shown you. Then we do a bioinformatic analysis and integrate human genetic variation that we will get from the same project, that we get in the same way from the work that I've told you about at the very beginning, that is done by the University of Tartu. In addition to this network mapping, especially Marianne, also us a little bit, will do very extensive functional assays to understand what each of these viral protein is doing functionally, on what impact it has on the host cell. For example, does it lead to an increase in proliferation or cell death? Does it impact immune signaling? All of these things are very important. And then both the data from the network analyses and from these functional assays are being taken by our colleague in Barcelona, who has very advanced AI technologies that both bring together these perturbations for us to develop very specific molecular mechanisms that we want to follow up and that we want to really understand in depth. But these tools are also suggesting small molecules that likely impact what the virus is doing inside human cells, and may prevent some of these impacts that we want to, that we really don't like. For example, the development of asthma or of severe infection. And so with that, I've told you about the first parts of what we're doing in this consortium, how we want to identify these risk factors, and how we want to bring them together on a molecular level through a network analysis. And admittedly, this is very detached from what we look at in terms of biology, from what's happening inside the cells. And now I'm handing the word back to Marianne, who will tell you how we bring this back into the cell level of individual cells and study specific mechanisms. Marianne.
Thank you, Pascal, for this wonderful spiel on how we do the nitty-gritty on the systems at systems level, how the viral proteins interact with the host predisposition in later developing asthma. Thank you for the hands up. Are there any questions? Please do use the Q&A. I'm sure you have questions. I'll give you a little bit of a chance now to type something in, because it will help our discussion in a little bit.
Thank you for reminding.
Now, what we're doing in Utrecht is taking all the abstract information from these virus high-risk variants, so the open reading frames, the genes in virus, how they relate or how they actually have interplay with the host high-risk gene variants, to later development of asthma. And we do this by looking in organoids, so mini organs from the lungs that we grow from different parts of the lungs. And we look in these organoids, lung organoids, what changes when you infect with whole virus, RSV, or with a transfection of certain open reading frames, high-risk genes from these RSVs, in predisposing to asthma-related cellular changes. Because of course, these organoids don't develop asthma themselves. It's just a little clump of cells, but we do know which profibrotic features or other asthma-related cellular changes we need to look at. So we take the least invasive approach. We're making organoids by nasal brushing for now. This allows us to grow from basal progenitor cells expanded cultures. We can preserve these cultures. With these cells, we do air-liquid interface cultures. And these are the cultures actually that we mostly focus on for now. And we go on to make airway organoids. And these type of cultures we use to transfect viral genes or these open reading frames. Viral gene is an open reading frame from high risk or low risk variants. We do functional assays to look at the cellular changes in these organoid cultures from the airways. And in the future, we will be testing candidate drugs to prevent this change into a pro-asthma phenotype, to revert it, to go back to the situation the cells had before the viral insult. Should not forget our collaborators. This is really done in collaboration with people from the Hubrecht Institute. They really pioneered the organoid technology, and they're also continuously improving these models, and we really are very grateful for them to be part of our consortium as well. Now, examples from these airway organoid cultures are here. So we just took 3 donors, and all 3 give the really good results. We get the beta-tubulin expressing ciliated cells. So cilia are the hair-like projections on the surface of cells. So they really move like with this motion, the microbes and the debris up and out of the airway. So you cough it up. And this is really motored by the beta-tubulin. So this is how you stain the ciliated cells. MUC5A is the main mucin produced by goblet cells in the airway epithelium. And these are non-overlapping. We see them in bigger magnification. You now see these hair-like projections in the ciliated cells. And these are from the same, in a different color, the same dish of cells. We see the goblet cells as well by staining for MUC5A. Now, again, we here in the Clarity project, we really tried, the goal is to investigate the functional impact of host genetic variation. So predisposition in the population on RSV early in the infant stage induced functional perturbations. And we study this by doing genome engineering by base editing. We start out with one set of airway organoids from a healthy donor, and we introduce these asthma-associated risk alleles. And then in these cells or these clumps, we affect them with RSV. Or we transfect with plasmids expressing particular open reader frames, these genes from the high-risk RSV strains that we get from Madrid, from our colleagues. And on the other hand, from Eric Ebner in Estonia, we get this information from the host side. So this is an example of the work he's doing with many others. It's a paper in Nature Genetics on long COVID, but it illustrates our approach. So here we see a gene that's high risk, at least one variant that's predisposes in their case to long COVID. And we take the same approach, looking through all these individuals in the Estonian biobank who actually developed asthma later in life and who actually had severe bronchiolitis because of a viral airway infection at the infant stage. So this type of information from hundreds of individuals, really this is a rich source, we combine with our own approach that we look in the lung cell atlas. It's open source. We look at what asthma risk associated genes are highly expressed in the epithelia or in other types of cells in the lungs. So epithelial cells are, for example, the club cells, the basal cells, the goblet cells, and other cells in the lungs. Well, think of mast cells, fibroblasts, neutrophils. For example, here you have the situation there if certain gene is expressed much higher in epithelium than in the other cells of the lungs. For example, fucosyltransferase. So this is work that's being done in the Utrecht lab by Daan Pion, postdoc, and Dagmar Duizer, PhD student in the lab. They focus on this fucosyltransferase that's highly expressed in epithelial cells and not in the other lung cells, or much less. And this fucosyltransferase, it's really an important glycosylation, post-translational modification enzyme. It puts these fucosyl moieties on proteins expressed that are secreted or that are expressed at the cell surface. And this variant in this fucosyltransferase really relates to it affects the extracellular matrix, and also affects the epithelial mesenchymal transition that we know of cancer risk, but it's also important for production of certain cytokines. It also is important for pro-fibrotic changes. So if you have the EMT, epithelial mesenchymal transition, is, for example, important in wound healing. If you have an aberrant type of wound healing, that can be pro-fibrotic because you have much more scarring, and we believe that this scarring may actually predispose also to later asthma development. So the question we take in this Clarity project is, how does this variant in this fucosyltransferase somehow relay information from the RSV strains to predispose together to asthma development? So we do this by transfecting this open reading frame, this viral high-risk gene, we also look in the organoids, expressing again by CRISPR-Cas the high-risk variant of this fucosyltransferase. And we do functional assays, how these organoids are now changed in their function. Are they more stiff? What change do we see that are pro-asthma related? And in the end, of course, we have another 3.5 years in the project. We will be testing candidate drugs to revert these changes to the situation, the flexible lungs, from before the viral insult. So this is again a picture of these of the histology of these lungs, these airway organoids. And I think this is the moment that I would like to open the floor for discussion. We have been talking about RSV to asthma, unraveling the link, applying fundamental science towards preventive medicine. Please.
Thank you, Marianne. Thank you, Pascal. This was an amazing outset of what you are doing and what you promise us to do in the coming years. So thank you so much. While people think about their questions they have for you, I will start with a question from Aisha Jeredi. And she has a question that I also had, is that she says, it's an amazing approach and very essential to understand the network. I was wondering how you distinguish cause from consequence. And maybe Pascal, you start first, and then Marianne takes over.
Yes. Well, I think first reasoning here is that I'm not sure I fully understand the question. Maybe Louis, you can refine if I'm going
Sure.
off to a wrong tangent here. Yeah. If you do have,
sure, very happy. So, in some of these networks that you study or look at protein networks or host response networks, you look at the interplay between what is defined by the host, maybe genetically defined by the host, and what is defined by the virus, maybe the specificity of the virus. So how do you basically distinguish the host from the viral vector if you study one from the other?
Well, these are, I think part of the answer is that in the identification, we focus on one and assume that the underlying, that the other component is fairly random. I mean, there is an independence, there is an interdependence. But when we look at thousands of individuals that are infected by different strains, and we see a constant signal, then we hope for the start that this susceptibility factor on the host side, well, actually, yes. So to be honest, we cannot completely disentangle that. So there's certain randomization here. We have a lot of different genotypes that affect that. So if a certain virus emerges as, consistently leading to a higher disease severity, then for the first part, we assume that this is somewhat independent of the host. Not completely. It's not clearly not 100% disconnected, but there's a certain randomness in the host genotypes, and the same is true for the other side.
And let me give you an example, Pascal, to dive into this a little bit deeper. So the RSV is suggested to play a causal role in the pathway from to asthma, a causal role. Rhinovirus is another virus that has been suggested to be an indicator virus that induces viral wheeze only in those who are susceptible, so genetically susceptible to the development of asthma. Mm-hmm. So I also thought perhaps it makes sense for your Estonian genetic biobank study, not just to look at RSV-related asthma, but also rhinovirus-related asthma. And then you might see that there might be overlap, which is probably pre-existent genotype, and the other one is maybe related to cause.
Yeah, so that's exactly what those folks are doing. Eric, who Marianne showed on a slide, he's actually doing not only the association study for asthma and RSV infection severity, but he's looking at many traits that are peripherally or very closely related to the traits that we are interested in. He's also looking at SARS-CoV-2 and many different other relationships in order to disentangle the specific signal from something that is just generic to viral infections of the lung. But maybe, Marianne, you do have additional things that you want to say to answer this question.
Thank you, Pascal and Louis. I would say we take organoids from a non-susceptible host. We put in the host susceptibility locus, and we compare, do we see changes that are pro-asthma? And at the same time, we only take RSV in a normal host, not susceptible, and we look for changes. We take the high-risk open reading frame. We see more. So it's a bit of a grid. We can compare the effects of innocuous RSV. Not all have the high-risk variants, the open reading frames. And also on the host side, we take some families, some host genes predispose more to asthma development than others. So it's a bit of a game. Yeah, Pascal.
Yeah, so my question, I was kind of hooked on the identification, but ultimately we do this in experiments, and so there we have control over what we are doing. We take different variants that are coming from viruses that we classified initially in the discovery phase as high risk or low risk, and we see what are the responses, what are they triggering inside the cells. And then for some of the variants that we look at on the human side, we will engineer cells that have one variant or the other, and then we can see how the combinatorial will play out. And in that case, there's a very clear relationship that we can establish. And I think generally we are looking at, okay, the virus is coming as a perturbation from the outside, and then afterwards it's a consequence of that infection.
Thank you.
So Pascal, you explained that you have used a similar approach in your SARS-CoV-2 studies over the past years or during the pandemic to develop small molecules. Can you tell us a little bit about the hurdles that you expect in the coming years that you may see? Maybe that has to do with false positive, false negative discovery, or manufacturing of those molecules, or because this is new to me, but I'm fascinated by your approach to use these networks to develop small molecules. Over.
Yes. So I think this will be actually much better for a separate webinar where we should invite our colleagues from Barcelona who have developed the AI tools to develop these kind of things. But I don't want to leave you hanging and just comment on a few things. So some of the possibilities, some of the challenges, of course, are not every target that seems good and suitable, not every protein that seems good and suitable can be targeted. So there are these kind of issues about where can we have a small molecule against this anyway, how long is the time frame for this, and one of the hopes that we have for this project is that we may discover something that is already approved and for where manufacturing has been solved and not been solved, that we can then bring to patients very, very quickly. But here the general risk, or the general risk not, but this is a generally a very unlikely scenario. So we're hoping for this. We have our eyes open to that, but it's not certain. When it comes to the drug discovery, it's not only based. I mean, ultimately, the drug discovery that we do in this project is not only based on the networks. The networks is one piece of information, but then we use all the perturbation data. And what Patrick Alloy, who's doing this kind of this part of the work package, is doing in his lab is he has a huge database that brings together the entire literature information. He has perturbation data for many different types of diseases and also for many small molecules. And then he's looking for similarities between what the virus is doing, what we see in asthma patients, and perhaps what can be changed and affected by small molecules. But this is a very novel AI approach, and it's very difficult to say how often this pans out or not. I think it sounds extremely promising, but on paper, many things have looked good. And in the end, the proof of the pudding is in the eating, and we don't know yet.
Thank you. Thank you. I like these AI helps of generating new molecules. I'm sure that they will be of help. So Marianne, there are several people with questions about your organoid model. And I will start with the first one, which is intuitive. Do you use baby organoids or organoids derived from epithelial cells from the airways of babies?
Yes, thank you for this question. We're going to compare in the project at later time the changes that we find with, organoids that we grow from babies as well that are infected with an airway virus. So, of course, we know that the vaccines against RSV now prevent a boatload of RSV babies entering our intensive cares. But, we think we should be able to find some, or we use historical samples to grow organoids from the airway samples from acutely infected RSV infants. At the same time, we also plan to make organoids from children or adults who already have contracted asthma to compare the features that we think are pro-asthma with what we induce by expressing high-risk operating frames from RSV, and also the mechanisms that we think are associated with later asthma development. This is part of our setting up the systems.
Thank you. Thank you. And for the people at home, feel free to use the thumb up button for the questions, so that helps me prioritize which questions to ask. So another question for you, Marianne, is the use of immune cells. I know this is super important because epithelial cells on their own, in the context of a virus, basically do not exist. It's always the interplay between the virus, epithelial cells, and the immune system. But it's really hard, isn't it, to bring in immune cells? Can you tell us about your approach there?
Yes, thank you for this question as well. So as I started in the webinar, we started out from Th17, IL-17 production. This is from 2013. That work that Louis and myself did back then. And that's a CD4 T cell producing IL-17. We know the IL-17 production mobilize neutrophils to move into the inflamed lungs that really make these lungs completely packed with all these inflammation cells, fluids, cytokines, the whole gamish. Now, in our project, we plan to take, well, we're happy to have donors who will provide us with T cells from the blood that we grow. We also, well, that we take, and we take the organoids from the airways, from the tube, the breathing tube from the babies who are intubated. And from the same donors, we do the co-cultures of CD4 T cells with the airway organoid cells, the epithelial cells, to look at how the interplay of these two cell types actually, well, maybe polarize the CD4 T cells to become more Th17-like or induce more production of IL-8. Anything that we think that we get a handle on, that we know increase the chance of becoming more, well, a phenotype of asthma. Does this answer the question?
I think so. Very well explained how you bring in the immune cells. So thank you very much. So before we go towards the end of this webinar, there might be still room for one question. We'll see. Is for you, Pascal. Maricho Caballero discusses that RSV is associated with different wheezing phenotypes. And I think he points to the fact that some of the wheezing is transient and the other one is persistent asthma. So different asthma phenotypes, if you will. Do you have the power or the intention to distinguish these different phenotypes within your analyses, either the genetic cohort or your networks, or maybe Marianne within her organoids, but that seems to be a little bit more complicated to me. Pascal.
Yeah, so my question there would be, so as you pointed out in the introduction, I'm not a clinician, and so I do not know how deep the phenotype recording is on Eric's side in Estonian database.
I don't either.
If we do have the matched data to really relate these two things. What I can say is that what we tend to do to use networks and what we find as a discovery tool is to generate the data, to also generate the functional data, see what each of these viruses and the viral proteins are doing inside the human cells, and then hopefully have molecular leads that can be linked and statistically linked to the phenotypes that we know are relevant for the different wheezing phenotypes, for example. Something that is more transient, or something that is more permanent, maybe involving more permanent changes in the lung cells. And so perhaps this can be related back to the fibrosis assays that Marianne is doing in her cell culture. And so, Marianne, maybe you do have something that you want to comment on here in this question as well.
Good point.
I think that pretty much covers it. I'm not sure what to add.
Thank you for demonstrating the links between various parts of the project. That's really the beauty of Clarity, that it's all so well interlinked. I've seen so many consortia, each going his way, and you really make it a concerted effort. So thank you again for doing this. Maybe next time we will hear others from the consortium. I hope it will be as good as it was today. With that, I thank the two speakers of today. I would like to thank the audience for being here and participating in the discussion. It's helpful for all of us, the consortium and the foundation that is organizing the webinar, to have your evaluation form. What did you like? What would you like to see again? What would you like to be different next time? And then see you again in future. It will take a few months, but you will hear from us. Thank you very much and enjoy the day. Bye.
Thank you all. Bye-bye.
Thank you.
Thank you.